These are notes on meiosis, or the making of unique gametes. Gametes are also known as your sex cells or your sperm and eggs. So we've talked about how every cell in your body has the exact same DNA, and it's just which genes are repressed or expressed that makes the cells different. And that's true for every single cell in your body, with the exception, because biology, there are always exceptions, of your sex cells or your sperm and egg cells. So if you are biologically female, every egg in your body will be genetically unique from every other egg in your body. If you are genetically male, then every sperm your body produces will be genetically unique from every other sperm your body produces throughout your lifetime. And so this is how we get such diversity even within human families. So if you are genetically 100% genetically related to a sibling, and what I mean by 100% is you and a sibling share the exact same biological mother and father, you'll still notice if you look at a picture of the two of you, even at the same age, you may look very different. So while many of us can look to our own families to see genetic differences between 100% genetic siblings, I actually really like this picture of this family because it starts to get us to question our ideas about race. So race is not actually related to biology. It's a societal thing. So when people say things like this race is better at this or this race is better at that, they're not drawing on biology. Those are societal constructs made to get people to believe things, really. It's sort of propaganda. So a nice way to think about it is race isn't real. It's made up by humans, but it controls a lot of what happens. And so that's one of the reasons we need to really fight against embedded racial differences in our society. So these two girls in the center here are actually twins. This is their biological mother. She's white, and this is their biological father. He's black. They're fraternal twins, which means that they came from two different eggs and two different sperm. So fraternal twins are no more similar or different than any other two siblings with the same biological parents. So what happens is in her uterus, there were two completely separate eggs that got fertilized by two completely separate sperm, but the eggs were from the same woman and the sperm were from the same man. They just happened to have the same birthday. Identical twins are a little different. That's one egg and one sperm, and then that kind of group of cells at some point before they started to differentiate split into two little groups of cells, and each one developed into a non-unique fetus. So identical siblings actually share all of their DNA. Fraternal twins share just as much as any two siblings with the same genetic parents. That is to say that all of this difference that we see is the result of only 0.1% of our DNA. Any human shares 99.9% of its DNA with every other human on Earth. And so that's also gets at this sense is there's so much more that goes on with us than these differences that we like to see and kind of focus in on, right? All of us have the exact same genes when it comes to regulating mitosis, for example, and we don't even think about that, or making a heart, for example, or making hemoglobin to transport oxygen around our bodies. I also want to talk really quickly about family being different from biology. We're going to talk about this in later in the unit as well. But I just think it's good to keep in mind that some of us live with our biological families. And so what we call family all kind of blends in together. Some of us don't at all. Or whoever gave us a sperm and egg that gave us life, we may or may not even know. They may not be part of what we call family at all. And then a lot of us live with kind of a blending of the two. And so I do just like I kind of want to call out that race is something that we've kind of made up, right? Simply because the person you call mom is not who your biological mother is doesn't make her any less of your mom. So family in a really good way is also a societal construct, which is really nice for us very social creatures. I want to give you some new vocabulary. These are diploid versus haploid cells. So diploid cells are cells that contain 2 copies of each gene, one from each parent. So up until this point, we've talked about how humans have 46 chromosomes. It's actually 23 pairs of chromosomes. So one of each pair, just like a pair of shoes, one of each pair comes from your genetic mother and one of each pair comes from your genetic father. And that is true of every body cell, or we call those somatic cells. And so those 46 come in 23 pairs. Your sex cells, on the other hand, are called haploid cells. Haploid cells, again, also called your gametes, contain one of each copy, one of each chromosome, or one of each pair of each chromosome, so that it can pair up with another sex cell, another sperm or egg, to get a full complement of the 46 chromosomes, right? That makes sense a little intuitively. Each sex cell has to have a specific 23 so that the final result, the zygote, has the right 46 instead of like 4 of chromosome 2, but none of chromosome 4, right? If you want the same 46 chromosomes going forward, then 23 from one pair and 23 from another pair have to pair up. Okay. So it's kind of like, again, if you think about the fingers, if I want a cell that has 10 chromosomes, 2 thumbs, 2 pinkies, 2 ring fingers, etc., then each of my sex cells have to have one of each of those. Okay. So we'll look at how that works now. This is the typical human genome, genome meaning all the genes we have. And so you can see there's these 22 identical pairs. We'll talk about X and Y down here in a second. And the banding is a chemical tracer to show where certain genes are. You don't need to worry about that. But you can see more or less these all look identical as far as each pair, right? If you look at pair 3 here, those pairs are about equal in length, where they have some dark bands and some light bands. Those more or less overlap. You can see, again, DNA's flexibility here, like with this chromosome here in pair 10. You can see some chromosomes are a lot shorter than other chromosomes. And so if you're looking at this, the way it works is that one from each pair comes from your biological mom, and one from each pair comes from your biological dad. Now, there's no rhyme or reason. Your cells don't know which one came from which parent originally. Your cells just know I have 23 pairs and they all have doubles. Now, there may be some differences in some of those doubles, and so we'll talk about that. It's kind of like if you have a pair of shoes, but one shoe has a smudge on it. Or one shoe maybe has different color laces from the other shoe. You did that either intentionally or that's how they came or whatever. Maybe one shoe has an insignia that the other shoe doesn't have, but basically they're the same pair. Then, down here, we have our X and Ys. So if you are genetically female, you actually have 2 X chromosomes. If you are genetically male, then you have an X and a Y chromosome. So that 23rd pair is different if you are genetically male versus genetically. As I said at the beginning of this video, this process of making the gametes or making the sex cells is called meiosis. These are the stages of meiosis. Do not write them down. You are not responsible for knowing all of them. Depending on what website you look at, some may have ones that are slightly different from others. Like, for example, some may have a prophase 2 right here, and some might not have the prometaphase. At any rate, you don't need to know all these stages. We're only going to talk about a couple. So what I do want you to be aware of is here is one cell with 6 chromosomes. That's an interphase, the cell just doing what it's doing, making its proteins, growing, repairing things. That's just the cell being the cell, right? Then down here, though, at the very end, we end up with 4 cells, each with 3 chromosomes, okay? So that's a big key. If we remember back to mitosis, it would have been starting with 1 cell with 6 chromosomes and ending with 2 cells, each with 6 chromosomes that were identical. Here we start with one cell with 6 chromosomes, and we end up with 4 cells, each with 3 chromosomes. And they're all unique, which also means that somewhere along the line, right, we've gone from 6 chromosomes here to 4 times 3 is 12 chromosomes. Oh yeah, our chromosomes have to double, right? So that's, again, that happens before meiosis starts. All of the chromosomes replicate. So instead of one cell with 6 chromosomes, before meiosis, we have one cell with 12 chromosomes. All those chromosomes have replicated. Now, through all of these steps that we're not going to focus on, we're going to end up, though, we now have our 12 chromosomes, so we can now get 4 cells, each with 3 chromosomes. We are going to focus on 2 phases. One is prophase or prophase 1, which is what I call it, because a lot of stages will have a prophase 2. And the other is metaphase 1. And we're focusing on those because those are the 2 steps that really ensure that all of these gametes down here are unique. And that overall is the point of meiosis, to make unique gametes. The first thing happens in prophase 1, and that's a process called crossing over or recombination. What happens is, remember I mentioned earlier that even though these chromosomes are the same as far as being pairs, there may be a few differences. And we can now understand, for example, maybe on your father's chromosome that you got, you had a functional freckle making, or let's go with PTC. You had a functional PTC making gene, but from the chromosome you got from your mother, you had a non-functional PTC tasting gene. And, you know, there's no reason to think that, like, it, you know, you would always get functional from both parents or always non-functional from both parents. There've got to be some times where you get a functional from one and a non-functional from the other. So that's where some of that little difference comes in, okay? So after the chromosomes have replicated, which again is back in interphase during the S phase as usual, we now have doubled the chromosomes. So in here, they're color-coded to be like the blue chromosome came originally from a biological mother and the red chromosome came originally from a biological father. But they've replicated, so we have 2 of each one, right? This is actually 2 chromosomes. Those little X's you see shouldn't so much be like that. It should be more like that, where here's one, here's the other, okay? So keep that in mind. This is 1 chromosome here, and it has replicated to get 1 chromosome here. And it's identical, right? And then these letters are whether a gene they're just different versions of the gene. Don't worry right now about functional or non-functional. But here we have like capital A, and so it replicated also as capital A. But here on the father's chromosomes, that's a different version of the same gene, okay? For right now, we'll call big A functional and little a non-functional. Doesn't matter. We'll get into this in more detail later, okay? Now, what's interesting, though, is because these are actually 2 of the same pair, even though they have slightly different parts, they can actually swap pieces of their genetic material with one another. That's called crossing over, because the chromosomes actually cross over one another, or recombination. They recombine where the genes are. So instead of getting, in the end, 2 chromosomes that look identical to this blue one and 2 chromosomes that look identical to this red one, these pieces down here swap with one another, and you actually get 4 unique chromosomes when it comes to what gene versions or alleles are on each chromosome. So we have this 1 chromosome here, which is big A, big B, big C. This second chromosome here, which is big A, big B, little c. A third one here, which is little a, little b, big C, and a 4th one here, which is little a, little b, little c. So instead of having 2 pairs of 2 identical chromosomes, we have 4 unique chromosomes, but all belonging to the same set. They're called homologous chromosomes, if you want some definition on that. Or some, not definition, but some vocabulary on that. So that's the first way that we can get some diversity in here, is we can literally swap pieces of chromosomes. The second way we get diversity in our gametes is something called independent assortment. So this happens in metaphase 1, and it's how chromosomes align, and that determines which gametes you can get in the end, okay? So let's take a look at this cell right here, right? So this has 2 sets of pairs, right? Meaning 4 of each, because the DNA has already replicated. And so you can kind of see these 2 are part of one pair and these 2 are part of another pair. You can tell because of the length. There's also some little genes on here, like here's the R gene, and that matches up with this R gene. Here's the Y gene, and that matches up with that Y gene. Now, there's 2 possible arrangements. You remember metaphase is the one where all of the DNA lines up. In this case, though, instead of one chromosome lining up with one chromosome, those homologous chromosomes are going to line up with each other. There's not a good way to do this with fingers unless I had somebody else's hand and we both had pointer fingers, and I don't. There's 2 ways that they can line up. Now, remember, we said before that the blue chromosomes originally came from your biological mother and the red originally came from your biological father. Your cells don't know the difference, though. So they just line them up any which way as long as homologous chromosomes are lining up with each other. Because again, similar to, you need them to line up in mitosis so that you get one of each, right? They got to line up with their homologous ones so that, again, you get one of each. So they could line up this way with all the mother's chromosomes on one side and all the father's on the other, or they could line up this way where it's going mother, father, and then father, mother, right? But that means then once those split, in one cell you have all the mother's original, and in one cell you have all the father's, whereas in these cells, you have differences, right? These are now different. So now when they split again, they've got to split again if you want one of each chromosome in each cell. Here we can see there's a lot more difference, right? We end up, in this case, if we've taken this path, we end up with these 2 gametes. But if we take this path, we end up with these 2 different gametes. And imagine that's what we're getting with 2 pairs of chromosomes. Imagine what we would get if we have 23 pairs of chromosomes, which is what we have. Tons and tons of diversity there. And again, the main takeaway here is meiosis makes diversity. So in summary, meiosis goes from one diploid cell, a regular body cell that we all have that has the exact same DNA, and you end up with 4 unique gametes in the end that are haploid, one of each chromosome. And so this diagram can kind of show you how you could get some diversity when you've got both crossing over and independent assortment. So as long as in each cell we have one chromosome that has the F and the T gene and one chromosome that has the M and the A gene, then we can get all 4 of these options, and we could even get more, right? We could have one that has this chromosome here and this chromosome here, right? That's nowhere in here. So that could even be a 5th possibility. And then again, if this is the diversity we can show with 2 pairs of chromosomes, we have 23 pairs of chromosomes. We have 30,000 genes, right? This is 2 pairs with 4 genes. That's what gives rise to all this diversity. And that's how, no matter how many children a couple might have, they're all going to be genetically unique. Of course, with the caveat that any human is only 0.1% genetically unique from any other human. But you take my point. All right, so please let us know in class if you have any questions, but this is how you make your sex cells.